Okay, we're going to start getting into tissues and how tissues support plant growth. Now, we've already kind of talked about tissues a little bit, and so some of this should be reviewed, some of it will be new, but I think going over it multiple times will only be beneficial. So, we're going to start talking with meristematic tissues. Those are the tissues that are supporting plant growth, right? They're the ones that are able to undergo changes into other cells and able to allow the plants either grow tall or grow wide. So for this video, we're going to start talking about defining and describing meristematic tissues, identifying the structures and functions of meristematic tissues, and then explaining the role of meristematic tissues in growth patterns. So to talk about tissues, we kind of need to back up and describe what is a tissue. Well, we've already talked about cells, cells being the fundamental unit of life, different cells performing different functions. Tissues are just one layer above that. They're clusters of cells that have similar functions and tend to work together. So they're working together trying to perform some sort of shared function to them. You can see in this picture here, this is a picture of the surface of a leaf, and this is made out of dermal tissue. So you have pavement cells that are working to protect the leaf. You have the stomata surrounded by the guard cells, which are allowing gas exchange, and together those are making a tissue. They're performing a similar function of regulating gas exchange at the leaf surface and protecting the leaf from desiccation and damage. So to kind of categorize our discussion for the week, here's a little diagram talking about plant tissues. Now, we have plant organs, tissue types, tissues, and cells. And so we're going to kind of work for this video up in this top part here, but I'll kind of go through the whole thing right now. So plant organs generally live leaves, stems, and roots. You can divide those up in two major tissue types: meristematic, the tissues that are rapidly growing and dividing, able to undergo changes into other cell types, and permanent tissues, mature cells that are carrying out specific functions. So we're going to talk about permanent tissues later, but when we talk about meristematic tissues, we will be talking about their role in supporting the permanent tissues. And so we can't completely disconnect the concepts. So for meristematic tissues, we're going to be talking about three different types. Apical, those are at the tips of shoots and roots, are allowing the plant to elongate. Intercalary, the ones that allow the plant to grow and lengthen even though the tip might be cut. And lateral, the ones that allow the plants to grow wider. So what is meristem tissue? It means divide into parts. It's tissues that are going to be undergoing rapid mitosis and then differentiation to support the growth of the cell or the plant. Now, we're going to be talking about primary growth and secondary growth quite a bit. Primary growth is the initial growth period of the plant. It's what's allowing it to grow longer. It's what's differentiating the tissues at the very beginning. And then secondary growth is the ones that growth that allows it to grow wider. All plants undergo primary growth. Not all plants undergo secondary growth. For example, if you're growing for just a very short period, maybe a couple months before you die, like a lot of annuals do, you don't really need to grow wider. You care more about getting up tall, throwing out your seeds, whatever it might be. But if you're living for multiple years, secondary growth becomes very, very important because you need that structure and stability. And so when we're talking about primary growth, we're talking about the tips of shoots and the tips of roots. Lots of cell division here in the shoot apical meristem, and lots of division here in the root apical meristem. Now, secondary growth, we're talking a lot about woody structures, like this one here. This is a second-year dicot. You can see it has 1, 2 annual growth rings, and then it's going to be widening out as it gets older and older and older. That's secondary growth. Now, meristematic tissue gives rise to the beginning 3 primary meristems. So the shoot apical meristem here is going to grow out. It grows these leaf primordia here, and it's going to give rise to 3 different types of primary meristems. Those are protoderm for dermal tissue, procambrium for vascular tissue, and ground meristem for ground tissue. So here's a little diagram that we're going to kind of follow. We have shoot apical meristem at the top, and we have another one later on for root apical meristem. So the shoot apical meristem, which is this piece, this dark spot right here at the tip of the shoot, will differentiate into procambium ground meristem, and protoderm. The procambium will then differentiate into primary xylem. Remember, that's the one responsible for carrying water from the roots to the shoots, and primary phloem for carrying the sugars wherever they need to go. It'll then also differentiate into fascicular cambium and eventually vascular cambium, which will differentiate or produce secondary xylem and secondary phloem. So we're going to follow this pathway in more detail in a second. Then shoot apical meristem can also turn into ground meristem, which will make pith and cortex, and eventually the cork cambium and phyloderm and cork. So the outside of the plant, inside of the plant, outside the vascular cambium, and inside the vascular cambium. It'll also turn into protoderm, the dermal tissue, and eventually into the epidermis. So here's our shoot apical meristem. This is the stem of a coleus stem tip. When I'm saying stem tip, think about, we look at this plant here, this little growth that's happening here. Think about the tip of that. That's what you should be thinking about when you're talking about shoot apical meristem, the tips of those growing shoots. So right here, ignore this arrow, that's just from the microscope. This arrow here, though, is pointing at this dark spot. That dark spot indicates rapid mitosis, rapid cellular division. So the plant's rapidly growing in that direction. Now here's some leaf primordia, and here's the beginning stages of leaves. Now, when we start to look at the leaf in detail, we can look at different layers. The outside layer here is now protoderm. Think about that as the first skin. It's the meristematic tissues that's going to develop into the dermal tissue. So we have protoderm on the outside, and then the inside you can already start to see this dark line forming. That dark line is the procambium. That's going to turn into the vascular tissue. So it's the early vascular cambium. And then in between the protoderm on either side and the procambium in the middle, you have these other tissues called ground meristem. It's going to produce the ground tissues, the sclerenchyma, the parenchyma, the collenchyma, those tissues there. So when you look at a stem tip, you should be able to differentiate the shoot apical meristem, abbreviated SAM, the protoderm, procambium, and ground meristem tissues. Now, when we're looking at root apical meristem, it's a pretty similar story. You have protoderm to epidermis, ground meristem to cortex, and then you have procambium to primary xylem and phloem. But then it also goes down to the vascular cambium, secondary xylem and phloem, similar to last time. But it also turns into the pericycle, which will then turn into cork cambium. So the cork cambium here differentiates a little bit. So if we go back here, the cork cambium came from ground meristem tissue, dividing from the cortex. Oops, wrong way. And if we go over here, it develops some procambium pericycle into the cork cambium. So rather than the ground meristem, it's coming from procambium. So just different developmental pathways. So here's the tip of an onion root. So we're looking at the onion root because that's what the tip of an onion root because that's where it's growing. Down here we have the root cap. So the root cap is a protective barrier for the root, allows it to push into the soil without incurring too much damage. And right past the root cap, you have rapid cellular division happening. So that's the apical meristem of the root. So rapid cellular division, and as the root continues to grow, this will differentiate into the three primary meristems. You have the protoderm on the outside, just inside the root cap in this picture. You have the procambium, the dark line starting to form in the middle. That's the early vascular cambium. And then the ground meristem again is before the vascular cambium and the protoderm. So it's right in between those two. This should be fairly easy to remember. Protoderm, skin, the outside. Vascular tissue on the inside, the straw being protected to funnel nutrients. And the ground meristem filling in the gaps. So that's the primary meristems. So then we're going to start to go to the lateral meristems. We talk about apical meristems, so the tips of the roots and the shoots, and now we're moving on to the lateral meristems. So these are the ones that are allowing it to grow wider and thicken. Remember, not all plants have this. Mostly dicots and gymnosperms. But monocots don't really have secondary growth like this. So secondary growth is going to allow to increase in girth. Now, this works in several ways. You need two different meristematic regions. You need one for the vascular cambium And then just exterior to that, you need the cork cambium to produce the bark, basically. And it depends on if it's in the roots or the shoots. So in the roots, it's forming from the procambium and the pericycle. In the stems, it's forming from the procambium and the adjacent parenchyma. Now, these are going to produce, the lateral meristem will produce the secondary phloem and xylem. The primary phloem and xylem is from the primary meristem tissues. The secondary is from the lateral meristem tissues. So we're going to kind of go into detail on these pictures. I'll zoom in here some more. So here's a zoom in on a dicot stem section. Now, if you start here in the middle, this is the pith. This is ground tissue. It comes from the ground meristem. Now, on the outside here, so here's the vascular cambium. Anything exterior to that is ground tissue, is considered cortex. So cortex, pith are both ground tissues, but the pith is inside the vascular cambium. The cortex is outside the vascular cambium. That's different than it will be in monocots, and we're going to go into detail in stem structures and differentiation between taxa in a subsequent week. But the cortex and the pith are just simply the ground meristem separated by vascular cambium. In monocots, it's either referred to just as ground tissue or just cortex without the pith. Now, when we're talking about vascular cambium, we're talking about this region right here. Oftentimes only one cell thick, just right around this outer ring here. And notice just inside the vascular cambium is the xylem. So xylem sitting in here, right? All of this is xylem. And then just exterior to the vascular cambium is the phloem. So this being the phloem in here. So the vascular cambium divides the phloem from the xylem. Xylem, remember, conducting water up, phloem conducting sugars wherever they need to go. The reason why this divide is important is xylem is dead at maturity. Phloem is alive. And so the outside of the vascular cambium is the living tissue. The inside of the vascular cambium mostly is the dead tissue. We'll talk more about stems later on, but you can also see in this picture growth rings. So here's the first year secondary xylem, and here's the second year secondary xylem. And you can see early on in the spring, the xylem ends up being really large cells, and towards the summer, the cells end up being really small, which is why you can differentiate the growth rings. Large to small, then large to small again, meaning 2 years of growth. Now, on the outside here, so we have the pith, xylem, vascular cambium, remember, which is giving secondary growth. It's going to move outwards, continue to lay down xylem on the inside and phloem on the outside. It's going to keep expanding outwards, and on the outer edge here is the periderm. Now, the periderm is composed of several different pieces. It's composed of phyloderm on the inside, cork cambium in the middle, and cork on the outside. So that cork cambium is allowing the outer region of the stem to grow as well. Here's a root for comparison. So in the very center, we have the primary xylem. Remember, that originated from the primary meristems. And then the secondary xylem, which originates from the lateral meristems. So down here, we have the vascular cambium. Inside of that, secondary xylem. Outside of that, secondary phloem. And then we have periderm again on the outside. So vascular cambium, xylem inside, vascular cambium, phloem outside. That pattern is going to be continuous through the roots and through the stems. So if we zoom in on the oak root and we look at the outside here, we have the vascular cambium, secondary xylem, secondary phloem. Phloem on the outside, xylem on the inside. Now, just exterior to the phloem in this picture, you can see these cells here, and look at how thick their cell walls are. That's sclerenchyma tissue. That's phloem fibers. That's there for support and structure to protect the vascular tissue just on the inside. Then you have phyloderm on the inside, which is part of the periderm, the cork cambium, and the cork. Similar to how the vascular cambium works, the cork cambium will produce phyloderm on the inside as a way of storage and protection. and cork on the outside as a way of allowing for protection of the plant. And this replaces the epidermis in perennial plants. So cork cambium, and phylloderm are part of the periderm, cork cambium being a secondary meristem tissue. We then also have intercalary meristem. Not all plants have this. It's only found in the monocots. And a good way to think about this is say you have a pine tree and you cut the pine trunk in half. Will the pine tree grow back to its previous form? It won't. It won't grow taller than that where you cut it. It might grow some sprouts out that kind of reach up a little bit, but the pine tree itself will not grow above that. And the reason why is there's no meristematic tissues there to allow it to happen. But compare that with mowing your lawn. You mow your lawn every week or so, or whatever, however often you mow it, and it grows back. Why is it able to grow back, but the pine tree is not? It's because grass is a monocot, and it has intercalary meristem. So here's a node. Say this is a piece of grass. Here's a node where a leaf breaks off here. Just above that node is this region of intercalary meristem. So it's a region of rapidly dividing cells that will allow the plant to grow taller and taller and taller. Even if the top gets chopped off, there's more intercalary meristem below it to keep growing. So the intercalary meristem allows for growth even after being cut off a little bit. Okay, so just kind of recap this video. I know there's a lot of text, a lot of vocabulary in here. The key here is really to memorize the vocabulary and then memorize the functions. I always recommend you do that. So you need to know the meristematic tissues, the apical, lateral, intercalary, the primary meristem tissues, protoderm, procambium, grand meristem. You need to understand how that relates to the vascular cambium and the secondary meristems, and understand those pathways of different meristematic growth.